Hello, this presentation is for students taking Anatomy and Physiology 1 at Madison Area Technical College with Gina Piscatelli. In this online lecture, I will just review the four major regions of brain anatomy, so it's an overview. Also, I'll discuss cerebral spinal fluid, its production, as well as reabsorption back into the blood, and discuss the blood-brain barrier. The average adult brain weighs about 3 pounds and is the size of 2 fists. It forms from a hollow tube called the neural tube, which develops by embryonic week 4. This neural tube is shown in the picture on the left. As the brain develops and continues to grow in the fetus, the neural tube folds and forms outpocketings or swellings that give rise to the four main regions of the brain that are present at birth as well as in an adult. These four regions are the cerebrum, the diencephalon, the brainstem, and the cerebellum. The brain is protected by the skull, of course, and covered by 3 membranes known as the meninges. The 3 meninges, from superficial to deep, are the 2-layered dura mater, the arachnoid matter, and the pia mater. Notice that the spider-like extensions of the arachnoid matter secure it to the pia mater. The space between the arachnoid matter and the pia mater is called the subarachnoid space. This space contains cerebral spinal fluid that further protects the brain by cushioning it. Floating within the cerebral spinal fluid gives the brain buoyancy and decreases its weight. Also notice that the dura mater dips in between the 2 cerebral hemispheres, and this is called the falx cerebri. The picture in the upper right shows the 2 dural septa that exist. The falx cerebri, which I mentioned previously, is located between the 2 cerebral hemispheres, and the tentorium cerebelli is located between the cerebrum and the cerebellum in the transverse fissure. Another feature of the dura mater relates to the fact that it has 2 layers. In some regions there are large veins, called dural venous sinuses, between these 2 layers. The dural sinuses drain blood from the brain, which then returns to the heart by way of the internal jugular veins. Besides being located in the subarachnoid space, cerebral spinal fluid is also located deep in the brain, within the 4 ventricles that develop from the hollow cavity of the embryonic neural tube. These hollow chambers are continuous with one another. The 2 ventricles in each cerebral hemisphere are called lateral ventricles. They are separated from one another by a membrane called the septum pellucidum. Both lateral ventricles have a channel that connects it to the 3rd ventricle. And the 3rd ventricle lies, it's narrow, and it lies between the 2 halves of the thalamus. And it's connected to the 4th ventricle by a canal called the cerebral aqueduct. The cerebral aqueduct lies dorsal to the pons and medulla oblongata. Its wall has 3 openings into the subarachnoid space called apertures. The lateral aperture and the median aperture are depicted on this diagram. The cerebral spinal fluid in the subarachnoid space provides protection and buoyancy, as I mentioned. It also provides an ideal chemical environment for the brain and the spinal cord. However, CSF is continually produced within the 4 ventricles by vascular structures called the choroid plexus, and the rate of its production is about 500 mils per day. but only 100 to 160 mils can be maintained within the ventricles and the subarachnoid space at any one time. Therefore, it must be removed continuously as well. This occurs by being reabsorbed into the dural venous sinuses through arachnoid granulations, which are labeled on the diagram. As CSF is continually produced, it first flows inferiorly through the ventricular system and then superiorly within the subarachnoid space and through the arachnoid granulations to reach the dural sinus. As more CSF is produced, a force is created causing the CSF to move. It moves, as I mentioned, from the choroid plexus through the ventricular system until it reaches the fourth ventricle. Then it moves through the apertures to the subarachnoid space. Now it moves superiorly in the subarachnoid space to reach the arachnoid granulations, which allow the CSF to move back into the bloodstream. It's then collected in a system of veins reaching the internal jugular vein, not shown on this diagram, and then travels to the heart. And the heart can pump out blood again, of course, through arteries, which eventually will end in the choroid plexus, where CSF can be made again. The choroid plexus, therefore, is where CSF is produced, and it is located in all four ventricles. The components of a choroid plexus include fenestrated capillaries and ependymal cells. The fenestrated capillaries contain slits, called fenestrations, that allow water and solutes to escape the blood. The ependymal cells are joined by tight junctions and regulate what substances can actually cross and enter into the ventricular space. In this way, ion and solute concentrations can be controlled to provide the ideal chemical environment or composition of CSF for the brain. So plasma leaks out of the capillaries, and ependymal cells form a barrier, allowing some substances to cross, like water, and also other solutes via transporters. Due to the selective nature of the choroid plexus, the composition of CSF is very similar to blood plasma, but it contains different ion concentrations and much less protein. Of course, CSF would contain glucose, oxygen, vitamins, and ions, such as sodium, chloride, and magnesium, but wastes would have to be transported across the ependymal cells into the blood. A disorder associated with cerebral spinal fluid production and reabsorption is called hydrocephaly. In this picture, you can see the normal size of ventricles on the left and the enlarged ventricles with excess CSF that occurs during hydrocephaly. The most common cause of enlarged ventricles is blockage of cerebral spinal fluid flow at the cerebral aqueduct. It's rare that CSF overproduction occurs, but it could occur when there's a certain type of tumor in the choroid plexus. So we have seen that the brain is protected by controlling the composition of CSF. This is known as the blood-CSF barrier, and it consists of the tight junctions between those ependymal cells and the transporters that are located in the ependymal cells that can regulate the movement of substances from blood to cerebral spinal fluid. But there's another barrier that protects the brain called the blood-brain barrier. It consists of tight junctions again, but in a different location. These tight junctions exist between cells of capillary walls in capillaries that aren't part of the choroid plexus. These tight junctions again regulate what substances the brain would be exposed to. So we should review the arterial blood supply to the brain to talk about these tight junctions or the blood-brain barrier. The picture on the left of the slide shows the extensive network of arteries that supply oxygenated blood to the brain. These arteries are the location of the blood-brain barrier. Their walls are the ones that contain tight junctions between the cells. Notice that the majority of the cerebrum is actually supplied by the internal carotid arteries and the vertebral arteries. The paired internal carotid arteries, shown on this diagram, but they've been cut, and the paired vertebral arteries, also kind of cut off at the brainstem, supply both the brainstem, the midbrain, and the cerebrum with oxygenated and glucose-rich blood. At the base of the brain, some communicating branches of these arteries form a circular connection called the circle of Willis. This union of blood vessels is referred to as an anastomosis. The advantage to an anastomosis is to provide collateral circulation to the brain tissue, ensuring that all regions of the brain are supplied by more than one main artery. So the blood-brain barrier, located within these arteries, is composed of more than just tight junctions between the epithelial cells of a capillary wall. It also contains a basement membrane lining these epithelial cells, as well as astrocyte foot processes. Together, these three components control what substances can cross from the blood and reach the cells of the brain. Notice in the diagram, for something to reach the brain tissue, it would have to move from the inside of the capillary wall, across tight junctions, across the pink basement membrane, and fit through the astrocyte foot processes. This blood-brain barrier is impermeable to some drugs, such as certain antibiotics, and it can complicate the treatment of neurological diseases. But it is permeable to, of course, water, glucose, iron, and the gases oxygen and carbon dioxide. It also allows alcohol, caffeine, nicotine, and some anesthetic pharmaceuticals to cross. In addition, there are two important neurological diseases for which we have treatment that can cross this blood-brain barrier. For manic depression, lithium can cross, and Parkinson's disease, L-DOPA, can cross. So the take-home message for the blood-brain barrier is that it can regulate and maintain a stable environment for the brain. It also has three components: tight junctions, a basement membrane, and the foot processes of astrocytes. And finally, another take-home message is how it is an advantage and how it is a disadvantage. It's an advantage to have a barrier between the blood and the brain to prevent the spread of infectious microbes from the blood into the central nervous system. However, a disadvantage is that certain medicines cannot reach the brain tissue because they can't cross this blood-brain barrier. That's the end of this lecture. Thank you very much.